Welcome to the 5th week of talks in the GCOOS Spring Webinar Series. The goal is to learn about the projects that the PIs funded under our new IU's 5-year award are working on. My name is Chris Simonello, and I'm the Outreach and Education Manager for GCOOS. I'll be moderating today's session, which is being recorded. Jennifer Vreeland will also be helping to monitor the questions that you can ask.
Yeah, this is for
either raising your hand or writing in the chat box. If we ask that everybody please remain on mute. And there'll be time for questions following each presentation. First up today is Dr. Eric Milbrandt. Eric is the director of the Marine Laboratory at the Sanibel-Captiva Conservation Foundation. In 2007, he established the River, Estuary, and Coastal Observing Network, also known as RECON, to provide data about the dynamics of the Caloosahatchee Estuary and the Gulf of Mexico. One reason the system has been so successful is that the information from RECON is summarized into weekly condition reports that get shared with academic, state, and federal partners, and these are served in a very timely manner, making them relevant to stakeholders. The range of data collected supports science focused on water quality, harmful algae blooms, and their effects on seagrass and oyster reef communities. Prior to Hurricane Ian, that pummeled Southwest Florida less than a half a year ago, RECON consisted of 9 real-time sensor platforms. The storm not only destroyed the platforms, the pilings that the sensors were located on, but it also brought about a 4-foot surge. Eric can enlighten us more about that, but damaging a lot of the operations and maintenance equipment that were housed in that building. The fact that Eric is speaking with us today is really a testament to the relentless work his team has been doing to restore capabilities. And today his talk is titled, Providing Real-Time Water Quality Conditions Through Harmful Algae Blooms and Hurricanes. And following your talk, Eric, I will introduce Dr. Chuanmin. So all yours.
Great, thank you, Chris, for the introduction, and thanks to all of you for being here. It's a really a pleasure to be able to have some normal functions to plan around, including sharing some of our work with you all today. So yeah, we are a nonprofit organization based on Sanibel and Captiva Islands, and we have a lab that's focused on regional water quality issues because of our connection to the Florida Everglades. So in Florida, we have an algae bloom problem. We were just talking about it on the before the presentation about how there's red tide in the east, there's sargassum, which Chuanmin will talk about, but we also have major blooms that occur in the lakes and freshwater areas that are sometimes, because of the managed system, discharged to the estuaries and coastal areas and cause big problems economically and in some cases from a human health perspective, especially when it comes to red tide, which is I'll talk about here in a minute. So these impacts have a really big influence on property values as studied by the Florida Realtors in 2015. I know that value is probably a lot more today than in 2015, but it was estimated that there was a $541 million reduction in property values that was related to transparency of the water. And those analyses were done using data collected by our organization on behalf of GCOOS in our observing network, which I'll share with you here in a minute. 1 in 5 jobs in our county is linked to tourism, so we're not an agricultural economy. We're a very strong tourism-based economy, which requires good, clean water and healthy habitats. So the other impacts that we know about, short-term health effects from aerosolization of toxins near the coast. We don't know as much about long-term health effects from some of the blooms, and end up with sick and stranded wildlife and fish kills. So it's something we value here, and the people that move here because of the environment in many cases. So one of my colleagues made this great graphic interpretation of 1870 versus today, where you replaced wading birds with millions of people, and this population growth was combined with a flood control plan for South Florida to increase agricultural production and decrease flooding and the risk to humans. There was a big hurricane and washed out the earthen levee around Lake Okeechobee and killed many thousands of people south of the lake. So after World War II, the area was further channelized, ditched, drained, and many of you have probably seen this graphic showing the natural flow versus the current flow, which has a lot of water coming from Lake Okeechobee, the Kissimmee chain of lakes, to both the East Coast and the West Coast, and not enough water going into the Everglades. There have been also major land use changes during that time, including the increase of not only agriculture, but suburban growth, developing, replacing wetlands, replacing things that used to store water and hold water, leaving us with a system connected to Lake Okeechobee that delivers water and pollutants. So, and you can't discount the impact of hurricanes. This year we had the first-hand view of that with Hurricane Ian and satellite imagery after showing the 500-year flood event for the Myakka River and the massive discharge of dissolved organic matter, which is correlated with full nitrogen and other pollutants caused from the overwash event. So these are very dynamic. This is a very dynamic system, even though it's highly regulated. So it's really important to have real-time information. And that real-time information, we do our best to synthesize and also to show to decision makers, to the management teams that are both the federal and state government, and then also the West Coast stakeholders, because there are sometimes operational options for water that can, it's really not benefit one group or another. Oftentimes it's who's getting the worst of it, and can we spread that around to other places, which is until the Everglades Restoration Project, which may be 100 years long, these projects that are coming online will help. But until then, we really are trying to manage with what infrastructure that's here. So I've highlighted in red the key items in the GCOOS strategic plan that we are addressing with our observing network, which, yes, I will talk about that in just a minute, kind of setting the table here. But definitely, I'll show you in this talk about coastal hazards and about how we're trying to maintain this ecosystem and the living resources within it at a state that's not degraded, which is very difficult. And then the human health and safety area, where with our collaborations with other labs on red tide specifically. I think we do a great job at outreach and stakeholder engagement. I'll share a link with you at the end if you want to receive more information about our organization. Water policy is one part of it, but it's a very, very important part. It's very high priority. So all of the data and the synthesis of the science, during the last decade is used to provide science-based recommendations, and that's really important to our organization. I won't really talk very much about the data management side of things, but we are currently collaborating with University of Florida and University of South Florida to look at the entire system from Lake Okeechobee to a watershed model, linking that to an ocean forcing model to better understand HABs, or harmful algal blooms, in the estuary. And with that, we've first collected data in 2007. So we now have a pretty robust 12-year data set at multiple locations within the estuary and the nearshore. So this is what GCOOS supports, and it provides support for, I would say, 3 of the sites for maintenance, all of the sites for the data management, and the sites that are here listed. We've actually added 2 more in the freshwater part of the system in response to recent algae blooms. And we call it the River Estuary Coastal Observing Network, or RECON. These are all the parameters that it measures. Of particular importance to us is salinity. Maintaining that salinity balance in the estuary it's easily controlled by flow, and we have really good models and good biological indicators that are responding to salinity in the estuary. So we use those a lot, but we are also, of course, very interested in hypoxic events, which we've documented and written about, and of course the harmful algal bloom signal through the optical sensors. So if you navigate to our site, this is kind of what you get: a really nice custom graphing page. You can plot parameters against each other. We got a small, well, it wasn't that small, but we got a grant from a local agency to help us build this site, and we're very happy with it. And of course, technology changes so fast, as Bob knows in particular, but others that are here, that we will need a new website soon. But for right now, it's good until we can make something better. As I explained before, we provide very detailed scientific observations to the Corps of Engineers and the Water Management District. The Water Management District also provides status updates for weekly calls, and certain times a year it's easy. Decisions aren't really being made. The system is we're trying to lower lake levels during the dry season, and we're trying to not, we're sort of holding water other times a year from the lake. But we also have a giant watershed that's twice the size as Lake Okeechobee. So there's a lot of things to consider, and what we try to do is show graphically how the data are related to our indicators, and also just looking at things like what happened with dissolved oxygen this week, and red tide, which is a very timely topic today. GCOOS has created this great project, which we're one of the holders of a couple of HAB scopes, and the HAB scope is a great tool to feed forecasts of respiratory irritation, and we're especially hard hit certain times of year, and the abundance data for the red tide organism, the dinoflagellate that causes the respiratory irritation and the toxin, is estimated from a clever program that looks at swimming behavior, and then we provide those live water samples and estimates as part of a large network, and it could only be done by an organization like GCOOS. But we're very happy to be a part of it, and it's a really great thing to share with our members, but our greater Sanibel-Captiva and Fort Myers community. So as I mentioned before, we're using the continuous data to also expand our research on harmful algal blooms, all the way from Lake Okeechobee out into the Gulf of Mexico. And these are our sites that are visited monthly, and we're collecting very detailed information about nutrient isotopes, and phytoplankton species composition with our collaborators, Dr. Ed Flips and Elise Morrison at the University of Florida. So this was a graphic representation of how the data, especially the temperature salinity data at first, but later on we're going to use the chlorophyll data to help the Army Corps develop a tool, if we can, it's ambitious, but to model all parts of this system, from blooms forming in the lake to the contribution of the watershed through a watershed model being done by David Kaplan, and then the physical model coupling those models together. Our role in this is to provide the continuous data and the very detailed harmful algal bloom and toxin analysis. And so this project is in year 2, and I've seen several of these models working independently, and we hope to be able to provide something that will be useful for decision makers. The nutrient sensor pilot project got off the ground in the middle of COVID, which, they did the training and we purchased sensors. The PI on this is Beth Stoffer. I noticed she was part of the seminar series, so I'll just advertise her as being our leader. We are collecting nutrient data continuously at one site, and unfortunately we lost nutrient sensors in the hurricane. So we're planning to deploy again the nutrient sensors when we get everything back together and out in the field for the summer in particular. We noticed with the dry period, the low detection, the concentrations aren't high enough to be detected, and so we just measure a lot of very low concentrations or zeros, and so we'll get that back up and active in the summer. So we are also collaborating with Moat, and I apologize, this isn't Jay Langen. This is his predecessor at Moat, Jordan Beckler, but we are trying to get this piling ready for a hyperspectral seawater scanning sensor that his team has been working on for a long time, and we want to get it out there because it seems like whenever it's deployed in the Gulf of Mexico, there's no longer red tide. But no, we really do hope to get that out, and we're working with them. We usually provide boat support, and we try and do some maintenance on our own equipment. So last little series of slides here is about Ian. I know there's if in the Gulf of Mexico, if you live near the coast in the last decade, we've all experienced some pretty major storm events. This one was a direct hit. We lost the bridge that goes to our island, and the inside of our lab had 51 inches of water in it. So the workshop that we maintain and prep sensors on the lower right was flooded with water, and consequently we've lost some of our tools, but we didn't lose any of our sensors because we moved them upstairs. We actually have a 13.5-foot high building, so most everything was safe that was at the lab. However, we left, I think, one of the most important things about having a real-time observing network and continuous sensors is capturing these events. And boy, we captured them. All right, we left everything out. We lost weather stations. Some of them were flooded with seawater, and some of them just broke because the wind speeds were so high. But in general, we fared okay. So the green stations are sites with real-time data that were being collected, and then we had a wave buoy, an axis wave buoy that broke free from its mooring, but we later recovered it. So the wind data, I mean, this is kind of sad because it didn't, the pilings end up breaking it, and not necessarily the sensors, but these are rated to 150 miles per hour, and we were measuring almost 100 miles an hour before things really started to go sideways. The wave heights that we recorded have to be the highest ever probably recorded in the Gulf, at least in the West Florida Shelf. I mean, these are incredible numbers. 23-foot max wave heights, 5 miles from Sanibel, which helps explain some of the surge that we were getting in the bay, which these are from the estuary or the bay. We were getting 5 to 6 foot surge, which is kind of what we got at the lab. But on the beach side, there were 12 to 18 foot surge, and some of that I think it was driven by the onshore winds and the wave heights and just the chaos. I'm glad I wasn't here to see. So yeah, so my summary slides, I think were, I hope that I can convey the importance of GCOOS to our organization and to this effort. Not only just the funding, but the support that we get is incredible. We need all these assets, and even if some of them are online at times, to help mitigate some of these impacts of things like red tides and hurricanes. We're working with Jorge and GCOOS on potential supplemental funding to help fix or replace some of the equipment that we lost, which would be a huge help. But the consistent funding that we've gotten has helped us maintain a relatively gap-free data set. And I know I'm going a little bit long, but this is like my last slide. So I think this platform of GCOOS is again, it's really important for what we're trying to understand and at least provide the best information possible for the coastline of Florida and for our community. So just want to thank Jorge and the previous executive director, Barbara Kirkpatrick, and all of the staff for helping stand us up after the storm, but also the support for our observations. And yeah, with that, I'm going to end my presentation, and we can either, I think we'll answer questions, or I can save them till later.
No, we have time for some questions, Eric. Great presentation, thank you. Remind folks you can raise your hand, or if you're a small enough group, you can come off speaker, or you can put it in the chat box. They're going to force me to be a very bad moderator and ask my own questions if they don't step up. Yes, Jorge, go ahead.
Thank you, Chris. Eric, thank you so much for that great presentation and that data around this the hurricane. It's unbelievable. So thank you for sharing. And something that I just want to say to the group that you most likely you didn't feel like had time to share everything possible today is that I was amazed also by the story that I've been following, Sanibel-Captiva Conservation Foundation and your group. How quickly recover and how resilient you guys turn out to be. So congratulations for that, and for anyone who have not been following that, the foundation case after Ian since last September, just go to their website and look into their newsletters. They tell all the amazing story of everything that happened after the storm and how the foundation recovered. So congratulations for that. It's a great example for all of us.
Yeah, thank you. And it's a really tells a lot about the staff and the leadership we have, and the support that we've gotten from private gifts, but also from our funding partners. So yeah, it's a mission that turned from one of protecting Florida's natural resources to one of helping our neighbors. Some cases, dealing with total losses of their only home. So I'm sure there are probably many stories like that, but yeah, we're glad to be able to help.
Thanks, Eric. We also have a question from Joe Polidoro in the chat box, and he asks kind of a two-part question. What stories aren't being reported or properly covered about your work? And another way of asking, what are the biggest stories in your mind based on what you're finding in the data over time?
Yeah, that's a good question. So I think the part of the story that is not a positive one is that we've lost quite a lot of seagrass and oyster habitat before the storm, and we're finding out that we're losing a lot because of burial and so on. So I think that the data that we collect show the impacts of discharges very clearly. It shows a very clear link between nitrogen loading and chlorophyll concentrations. So, those are things that we knew already. We not maybe necessarily advancing the greater science of it, but we certainly are having an impact on people's comprehension about what the drivers are and how complex these issues are. So I see another question that, yeah, the 52% of the lake water from Lake Okeechobee is coming to the West Coast. And the reason why that is, we don't have, we actually asked for water from the lake this time of year to help maintain a gradient of salinity in the estuary that benefits our indicator species like tape grass, which is a submerged plant, and oysters. And the politics around where water goes right now is incredibly hot and heated. The East Coast it should receive some of the water, and the South receives water. So those are the three areas. But the lake is high, and that's not good. So we're fine with the current percentage, but we also need to lower the lake level. So more water needs to go to the East Coast, to be frank. But those are very heated conversations, and no one wants to release an algae bloom into the estuary. We're all quite sensitive to that. So.
Thank you, Eric. Oh, there's one more question in there. You saw that. I could see by your reaction.
Yeah. Who decides? Well, many people, but the Corps of Engineers ultimately decides. But you can imagine that agricultural interests south of the lake, they're seat at the table. There are stakeholders from the East Coast and stakeholders from the West Coast. And the South Florida Water Management District is basically the consultant to the district, or the Army Corps, I'm sorry, and then the Army Corps makes the decision. But that's where our weekly, we do weekly reports, we have conversations, and we try and get the best available information in their hands. We're sort of saying right now there's a red tide, and we're still receiving most of the flows. So maybe not increase the amount going to the west. It's probably not a good idea right now.
Thank you, Eric. That was great. And it's really nice to have you back. It's inspiring to see the progress that you've made in that time. I know how difficult and the challenges that you were dealing with. So welcome back.
Thank you.
Next up, we have Dr. Chuanmin Yu. Chuanmin is a professor of optical oceanography at the University of South Florida College of Marine Science, where he's also the director of the USF Optical Oceanography Lab. He specializes in using laboratory, field, and remote sensing techniques to study algal blooms, oil spills, water quality, and global changes. Using the virtual antenna system he established to enable the downloading and processing of satellite data in near real time, his team has developed some really cool applications, including the Sargassum Watch system, the virtual buoy system to monitor coastal water quality, and IRIS, the integrated red tide information system. And Chuanmin, I'm not sure if you ever sleep. You've mentored more than 30 graduate students and postdocs. You've authored more than 300 peer-reviewed articles and book chapters. I know you served as lead editor and other editing jobs for numerous journals. a reviewer for many programs, and host to visiting scholars from around the globe. I don't know how you do it. That's usually something we read for folks that are getting ready to retire. We're very grateful that you made time to talk with us today. And the title of Chuanmin's talk is Near Real-Time Remote Sensing, Monitoring of Water Properties of the Gulf of Mexico. So all yours. Thanks.
Thank you, Chris. Can you hear me?
Yes.
Good. Can you see my screen? Yeah. I'm trying to put in slide mode. Wait. You see my screen in slide mode?
It looks great, but there, yes, now it's in slide mode. All yours.
Okay, thank you. Thank you very much. By the way, I do sleep a lot, at least 8 hours a day. I'm just lucky to have very good students and postdocs. They do a lot of work. Anyway, good afternoon, everyone. Thank you so much for attending this session. So today I'm going to spend 15 or 20 minutes to talk about our near real-time system to monitor water properties of the Gulf of Mexico using remote sensing. So what am I talking about? What water properties? There are many things. In the Gulf of Mexico, we have recurrent red tides or harmful algal blooms. And we now also have sargassum inundation events, sargassum beaching events. We have a coastal water quality degradation after storms, after other things. We have oil spills and other water quality events. So there are many things going on. But then why? There are two meanings of why. Why do we want to monitor and study them? And I don't need to say anything to justify that why. But why do we use remote sensing to do this work? That I want to justify. That's many years ago. The Gulf of Mexico Alliance designed a chart to guide the future plans, where to put buoys or monitoring stations. This is quite comprehensive in the Gulf of Mexico, and there are many more stations in the national estuaries, in 7 estuaries here, and the Tampa Bay is enlarged. That's just our hope, okay, our goal. But in practice, there's always a lack of funding. And Erica showed a very nice in situ network, in situ monitoring network. And he can tell you how expensive and how labor intensive. So we do want to complement our this in situ monitoring network with the remote sensing, because remote sensing does provide way more data. Here is one example. To the left, I show over the course of 17 years, from 2003 to 2019, the number of water samples collected by FWC and other collaborators from 17 years. So the number of samples is aggregated in each 0.5 degree grid. That's 50 kilometer by 50 kilometer. You may see, oh, that's a lot. There's 1,000, there's 100. But think about if you have 100 water samples within 50 kilometer by 50 kilometer in 17 years, then how many water samples do you have in that grid in just 1 year or in just 1 month? It's perhaps 1 or 0.5 or none. So that's very few. But that's still a huge effort, because you have to analyze every water sample under microscope. That's extensive effort, but still that's not enough. And in comparison, we have the data quantity or frequency from one satellite, the MODIS Aqua. Okay? So after excluding clouds, sun glints, many artifacts, we still have this many observations in each 0.5 degree grid. So the color, the color tones are the same, but the scale is different. So to the right, the green shows 10,000. To the left, the green shows 50. So we have way more data to play with the satellite if you trust them. Okay, so that's a big effort we have put, to make the data trustable. Then talk about HAB. So this is the difficult part. We have to make the satellite data trustable by people. Okay, we have to develop algorithms and validated algorithms. We have to develop products and have confidence in these products. So all those have been published in peer-reviewed publications. But here, I want to show you what we did beyond those points. We want to implement the algorithms and data products in a user-friendly way to serve the people. So that is really beyond research. So over the years, we have developed a virtual antenna system. So this is the virtual, but it's an antenna. It's similar to a real antenna. It downloads the raw data from satellites, then process to higher level data products using our customized algorithms, and generate these products in user-friendly format and put them on the web so everybody has access. And in addition, we combine the HiCom or FVCom model surface currents. So not only you have access to images, you have access to model outputs. And in addition to that, we have in situ data in the system. So we combine everything. So from those, we target this virtual antenna system to several subsystems, including the virtual buoy system, integrated red-tide information system, Sargassum watch system. And within all the systems, we use many satellites. So everything is automated. Okay, so if everything works and we are satisfied with the products, we don't need to do anything. But that's not the case. Frequently, the system fails because of many reasons. We have to continue the system, and also there are always artifacts in the data products we have to improve. That's what we are working on right now. And GCOOS has been funding us to improve the data products and sustain the system. Now, several years ago, one of my students recorded a sequence of mouse clicks to go through each individual subsystem, so I don't have to repeat. So this is the animation of this mouse clicks. So I will just play this recorded system and explain to you when the system plays by itself. Okay. So here you enter the address of our website. That's optics.marine, and you have a menu. And under this menu, you have the virtual buoy system. And once you are in this system, you have a lot of clickable maps. You have many maps in different regions. Click on one of them, you see different virtual stations. These are virtual stations. Click on one of the stations, you have a summary of water quality at this station, including SST, chlorophyll, backscattering, light availability, light and attenuation. If the current value is abnormal, it's color-coded. And you can also go to individual parameter to have a time series to show the annual variation, to show the seasonality and the trend. You can save the graph. It's automatic. Each time you click this, the graph is generated on the fly. Okay? You can see the weekly composite, weekly mean data, in this case SST. You can also see the ASCII data file. If you want to combine this data with your data to study a phenomenon, these are ASCII files of mean and the standard deviation and the number of data points used to calculate the mean. This is just like a buoy, real buoy. Okay? You have monthly mean and weekly mean. And plus, you have access to the images. And then to the left, you have a calendar. You can click on any day. Now we have a complete coverage for the past 22 years. So any day you click a date, click sensor, click data product, you have the image. You might say, oh, I don't know how to interpret, but there's a Google Earth button there. You click on that button, you bring the image in your Google Earth. You can navigate. Plus, you have the surface currents overlaid on the image automatically together with a color legend to tell what is the temperature. Click on the arrow, you can read the current, the speed, and the direction. Now, if you have something in the water, you can predict, oh, where is that something moving with this current? You can turn off the current and turn back on the current to see the image better. Okay, so that's the virtual buoy station, virtual buoy system. With the integrated red information system, is the same thing. To the left, we have menu bar. You go to the clickable map and click on the area of your interest. You have an image. Okay? You have different types of image. You click on the sensor. You click on the type of image you want. Then you can visualize the water property patterns. In this case, the solar stimulated fluorescence. So the greenish color means you have a lot of fluorescence. Likewise, you click on that GCOOS button, you bring the image to your GCOOS together with the ocean currents and the FWC water sample cell counts. Okay? So one mouse click will give you everything. That's why we call that integrated. Okay? So you can click on the arrow to show the ocean current. You can click on the balloon or the circle. That's the water sample value. Okay? And then you read the detail about this, who collected the data, what is the latitude, longitude, what is the data value. So all this, so that will give you better visualization of everything, the image, the cell counts, and ocean currents. Of course, this was made in 2017. Now we have made a lot of improvements to remove these stripes to have a better system. Now going to the Sargassum watch system, again, we go back to the menu. There's a Sargassum watch system there. And you have introduction, you have data access, you have a clickable map to let you click on your area of interest. In this case, that's the Eastern Caribbean, where we have the most Sargassum. And you can click the date of your interest, click on the cell type sensor and the data product. Okay. In this case, it shows the river plume. From the Orinoco, and you can click on this alternative floating algae index and click on the G button to bring that image to your Google Earth. So again, we have ocean currents overlaid on the image, and if you know how to interpret the image, okay, you can find the sargassum from this image. I know, perhaps you don't, but now we have better products, and everybody can knows how to interpret. Okay? So this by itself forms a short-term prediction system. If you find large sargassum rafts in a certain location, you know ocean currents, you can predict for the next day or so, maybe 2 days, but not more. So that's the 3 subsystems. All are available through our website and through a mouse click. So I think that stops here. Okay. Oh, boy. Wait. Did I stop here?
You have a few more minutes. Do you have any other slides that you'd like to share?
Yes. Yes, I have a few more slides. So
you're fine. Yeah.
Okay. So let me put in slide mode again. Okay. So I say now we have a better product. Now everybody knows how to interpret. So this is one of them. So we interpret the images for you. So anybody can say, oh, there's something in the ocean. So this is the animation of the floating algae density maps. So if I click on this, oh boy, here. Okay. You see, you can stop at individual frame and track the date, track the sargassum map. And that's our goal. We want to do everything in animation mode, so everybody has access to design your time window to design your area of interest so you can track things. Okay, and more than that, now we have a thematic maps of red tides. So every white color is a red, heavy red tide pixel. So within a week, you can see how the red tide of Charlotte Harbor evolved from just a couple of small patches into a very large continuous patch of Charlotte Harbor. So that's a thematic map, and we are working to implement this type of thing on the web. But right now, we do have this. This is from yesterday. So this is from yesterday's image, yesterday's ocean current, yesterday's cell count. You click the Google's button, you bring everything to your Google's. So you have the most current red tide information by through one mouse click. So here is my last slide to show a summary. So what is the GCOOS funding used for? That is useful to sustain and improve all these existing remote sensing subsystems, coastal ocean observing systems. So what are the areas of the GCOOS strategic plan? Does this work align? Coastal hazards, healthy ecosystem and living resources. Sargassum is definitely a habitat, so it's related to ecosystem and human health and safety. So both red tides and Sargassum inundation are related to human health. How is this work advancing observing and monitoring capabilities? Remote sensing provides a unique value to complement, not to replace, but to complement field data in both retrospective assessment and near real-time monitoring. And here I focus on near real-time monitoring, but if we want to go back to the past 20 years, we have time series data to support research. With that, I stop and welcome your questions. Thank you.
Thanks, Chuanmin. Always fabulous. The work that you're doing is always cutting edge and really exciting. I don't see any questions in the chat box yet, but if anybody has questions, you're welcome to raise your hand.
Good.
Everybody's mesmerized. I do have a question for you on your time series, seeing the evolution of the red tide from Charlotte Harbor all the way up. Are those time averaged over days, weeks, hours? How are those put together?
No, those are daily snapshots.
So each is a daily, okay.
Yeah, every image is just one snapshot, just like when you take a photo.
Okay. Jen, if you want to take down the presentation, I'll be able to see all the hands. Jorge, go ahead.
All right. Okay,
Thanks, Chris. I do have a question for Chuanmin. Again, congratulations for the really comprehensive presentation about everything you're doing, Chuanmin. I meant to ask on the Atlantic full basin scale. I mean, what is the extent of your modeling? Is it including from the Mid-Atlantic to Puerto Rico? Can you just remind us, I mean, what you're doing outside the Gulf of Mexico as well?
Oh, yes. So thank you, Jorge, for your question. With the satellites, we really have no geographic limits. Once we have research interest, or once we are funded to do something in a region, we can cover that region. So the Caribbean Sea is really, heavy in sargassum this years, and we are funded by NASA to put out a monitoring system for the Caribbean and even for the Atlantic to look at the sargassum. I'm not sure if I answer your question or you asked something else. I did not capture the first few words you said.
No, that's fine. That answered the question. So basically, you're covering the wider Caribbean, including Caribbean and Gulf of Mexico.
Yes, and Gulf of Mexico is also one of the regions. And the animation of the sargassum map is for the Gulf of Mexico.
Okay. Thank you.
Hey, Chuanmin, I was really interested in your summary of the October red tide event from this year, and that our theories about this the species and this algae bloom is that it starts offshore and it's blown in or transported in with currents, and then all of a sudden it intensifies near shore. But it appears that it intensified and formed near shore in this case. I wonder how often that is happening.
Well, this is a very scientific question. Okay. I can't say for certain, what exactly happened in last October. But it appears from numerical model, there was offshore water. First, there was upwelling, but the freshwater flux from inshore certainly added nutrients. So that way, that may stimulate the Carnium brevis growth. So it's more than one factor. What is difficult is to know the timing, because neither can tell a complete story. So we have to combine everything, and we have to validate the numerical model as well. Does the upwelling, which is known from the numerical model, does the upwelling carry the Carnium brevis cells from offshore deep water to the surface nearshore water? Then we need to know whether there are offshore deep water containing Carnium brevis, and that is a gap right now. We do know there are a lot of algae in the bottom layer of offshore water, but are those algae Carnium brevis or something else? Somebody, we need to measure those. But what you measure in the upstream of the Klaus-Sahachi River is super important to add to this story.
Thank you.
I think that kind of builds on the question that Joe also put in the chat box about what insights are you starting to see and capture with these data?
Oh, the what big story can I tell? Or
what insights are you starting to capture with these data?
With this data, we see an event can be impacted by many things, and also we can tell some events did not occur immediately after others. There's a lag time. The big story, and currently, we can tell the sargassum amount, is alarming. That is pretty big. And red tides, I think everyone in the Gulf of Mexico is familiar with the red tide, but not everyone is familiar with the big sargassum inundation event. So that is big. And this year it started early in the Florida Keys. So the lower Keys already started to see large amounts of sargassum in last week. And without satellite remote sensing, it is very difficult to tell ahead of time. And now we have a tool to tell even 3 months in advance. So there's something in the Atlantic, and eventually it will come to Gulf of Mexico and may go to Alabama or Mississippi. It's like monitoring and predicting hurricane track. So that's pretty big. And it is relevant to all Gulf Coast states, not just Florida. Alabama, Louisiana, Texas, they all have the similar things. This is just like red tides.
Thank you. And I guess this is a question for both you and Eric in terms of, because you said the sargassum is more isolated on the East Coast versus the red tide on the West Coast. But what about in places, especially Caloosahatchee, and where you have a lot of freshwater flow? Are you able to distinguish and start parsing out cyanobacteria versus red tide versus, these other algal species that are mixing using the current techniques?
Is this my question?
I think you both probably have a different perspective, because Eric's kind of looking at it from on the ground and in situ sampling, and you're looking at it from above with satellites. So I imagine there's different approaches going on.
Yeah,
Let me
I
go
can
first.
go ahead,
You go ahead.
So
Yeah.
one of the things that we've done is we've added PC sensors that help. So they're tuned. They're optical sensors tuned specifically to detect cyanos in the freshwater parts of the system, and that includes the upper part of the estuary. We haven't seen any visible like flock kind of blooms on the surface, but we are sort of looking at this summer being kind of the first year where we can look at chlorophyll data. We have samples with detailed phytoplankton information, and then compare that with the new PC sensors.
Okay, add to that, if cyanobacteria form surface scums, under calm conditions, wind is very low, they tend to float on the surface and form scums. We can detect them very easily from satellites. And most of the time, that's the signature of cyanobacteria. Other algae, even Crania brevis, rarely form scum. They do occasionally, okay, but it's rare. Okay. So that's a signature, almost signature of cyanobacteria. But once we can tell that, we can separate the others. So we can tell cyanobacteria and non-cyanobacteria. Now, if you keep asking, can you tell whether it's a diatom or Solanaceae or Crania brevis? Well, that's more difficult. For that, we need some in situ data to confirm what we see from satellites, especially in Chalet Harbor, in the estuary. That's very difficult from satellites.
Which is why the two of you are such a great pair talking today.
Thank you. Thank you for the nice arrangement.
Thank you.
Jen or Jorge, am I missing any questions? I don't see anything in the chat box. Any other hands? So we're coming up upon the hour. I want to thank our speakers, Eric and Chuanmin. I'm sorry, Jen, go ahead.
Nope, I was just going to say I don't see any more questions either.
Okay, thanks. Thanks again to our speakers. It's been great having you both here today. Learned a lot. I want to thank Jen for all of your communication support on the tech side, Nadine Slimak for her communication support, and all of you for joining us today. I hope you join us next week when we have Chad Lemke from the University of South Florida talking about glider missions that they're working on, and also Dr. Chuanmin Lee from LSU talking about the WAVE system, information system, WAVE's current surge information system for LSU. And until then, be well and have a great week.